Okay, so I know that we actually left off way past this point at active transport, but I want to go back for just a moment because I realized I forgot to talk about two things associated with facilitated diffusion. And the first one was with this carrier-mediated facilitated diffusion. So we'll just call it carrier-mediated diffusion for now because we know it's facilitated by this protein channel. So what I want to mention is this term that we use with these channels called saturated. And what this means is that these carriers can actually become saturated when all of the carriers, let's say for this example here, right? We have glucose. When all of these carriers are actually bound to that molecule glucose that they're transporting. And so we call that point the saturation point because in this case, right, the glucose transport would be at its maximum rate. So it couldn't move any faster. And I like to give the analogy of this as an assembly line, right? So here there's all these protein transporters that are transporting glucose across the membrane. And if you use the analogy of, an assembly line of workers and they're all, let's say, sewing a tag on a shirt, okay? If each person on the assembly line, right, has a tag and they're currently sewing that on the shirt, then that's your maximum rate for that production line, right? So that's the same case here. So we call that term saturated, and it's just kind of an important term I want to introduce because so it's called saturated. So because I want you to know that these carrier-mediated diffusion proteins can actually have a maximum rate when they're completely saturated with that molecule that they're facilitating the transport of through the membrane. Okay, so now let's go ahead and go on to the next slide here, which shows the channel-mediated facilitated diffusion. And what I want to talk about is there are really two types of channel-mediated facilitated diffusion channels. So one is going to be called leakage channels, which is basically what this one is here. Oh gosh, my writing is always so special on this. Leakage channels. My goodness, I'm really challenged today. So, and the leakage channels, wow, my bamboo just doesn't want to move with me. So that's a leakage. If you can't quite read my kindergarten writing there, that's a leakage channel. And so again, that's just where whatever your solute, in this case again, remember it's usually small polar molecules, especially like ions, are just going to leak right through. Okay, we're going to talk about this today. We're going to use this term today when we talk about resting membrane potential. So then, okay, you have another type of channel, and that is your gated channels. And your gated channels are just like it sounds. They're going to have a little gate on them here that can be activated to open and activated to close. So it'll have a little gate here. And so again, these channels will be controlled, and they'll be, they can be maybe inhibited by certain molecules. They can, be promoted to open by certain molecules. And sometimes they can, again, be the initiation for that opening of that gated channel can be something like the change in voltage in the cell. Okay, and so then that gated channel can open, allowing, again, those molecules, those solutes to get through. Okay, so I just wanted to mention that those are the two types of channels, leakage channels and gated channels. And again, gated channels can be controlled, by certain chemicals that need to bind to it to open the gate, or it can be an electrical signal, like, again, a voltage change across the membrane. So again, it can be triggered by, some type of chemical that has to bind to it, some type of solute that has to bind to it, or it can be a change in the electrical. Oh my gosh, this is If you can't read it, that says electrical. Again, gradient or change in voltage across the cell. So we'll talk more about that. Okay, so let's go ahead and jump ahead. I'm going to go ahead and jump back out here. And then we're going to go back to active transport. And this will make sense why I kind of wanted to jump back and talk about all of that once we jump into active transport here. So remember here, I think I kind of had mentioned before that active transport is the opposite, right, of passive transport. So active transport is where you're going to want to move something against its concentration gradient. Or you're going to want to move a lot of a substance into a membrane, into, either into or out of the cell, I should say. And that's what we call vesicular transport. Both of these, again, types of transport are going to require ATP to move solutes across the plasma membrane, okay? So again, oftentimes this is used because the solute's too large for the channel. So again, a lot of times we'll use vesicular transport sometimes for that, especially if you're gonna be moving a bunch of solute. Or again, the solute's not lipid soluble, it can't move right down, okay? And the biggie is, this is the big one, why we use active transport. And this is what I really want you to think of when you're thinking of active transport. And sorry, I flipped back to the other slide here because I wanted to show you this. Active transport is really going to primarily be used when a solute needs to move against its concentration gradient, okay? So again, you can think of when you're moving solutes down their concentration gradient, right? This is passive. It's kind of like rolling a ball down a hill. But if you need to move a solute against its concentration gradients, this is when you are going to need energy, and you're going to need that energy in the form of ATP, okay? So we're going to talk about lots of examples of this. So again, active transport is, of course, going to require some type of carrier protein, and when you talk about that solute, it's going to bind specifically and usually reversibly with that substance that's being moved, meaning that that solute will bind, and it's oftentimes very specific. So we're going to talk about like a sodium-potassium transporter. And so that transporter, that protein transporter, is going to be very specific for binding, sodium on one side and then potassium on the other side. So you see they're very specific. But again, then this is going to be reversible, meaning they can bind to it, undergo a conformational change oftentimes, and then release it to the other side. And then it can bind another one. So we'll talk about this. This will make a little more sense when I have the sodium-potassium transporter up. So some carriers, just like I talked about just a minute ago, some carriers can actually transport more than one substance. And so there are two types of carriers that can do this. There's antiporters, which are going to transport one substance into the cell while transporting a different substance out of the cell. So I'm going to give you the example of this in just a minute of the sodium-potassium pump. The sodium-potassium pump is an antiporter. If I can get my pen over here. Oops. Hold on here. Oh, my drawing, I'm so sorry. My drawing is just so terrible. Let me try this again. Sodium potassium pump, okay, is a really good example of an antiporter. And then symporters are going to be things that transport two different substances in the same direction. Oftentimes, it's using the concentration gradient of one substance to transport another substance. So we'll talk about that. But again, for these to be active transport, they need to be moving some type of solute, at least one of those solutes, against their concentration gradient from a low concentration to a high concentration. Again, you're kind of going uphill here, right? And this is going to require ATP. It's going to require energy. So we're going to talk about primary active transport, and then we're going to talk about how primary active transport can be used to set up to set up the cell to do secondary active transport. So let me talk about primary active transport. And this is the easiest one that most of you think of when you think of active transport. This is the one that requires energy directly from ATP hydrolysis. Remember, what's happening there is just the ATP, remember, has these high energy phosphate bonds, and what's going to happen is it's going to drop a phosphate bond onto that protein pump. That's going to cause it to undergo this conformational change. And again, I'll show you this in just a minute. And this is the easiest one to kind of study this with is the sodium-potassium pump. And the reason that we're going to talk about the sodium-potassium pump is that it's very important for establishing what's called the resting, or helping maintain, I should say, the resting membrane potential for our cells. So hopefully some of this is kind of clicking in from your old 181 days, right? And so when we talk about those sodium-potassium pumps, I'll show you in just a minute. We're going to pump sodium out of the cell and potassium back into the cell. And I'll show you this in just a minute when I have a nice diagram to show you that. But what's going to happen is then we're going to establish an ionic gradient by using this sodium-potassium pump, by pumping a bunch of sodium out of the cell and pumping potassium back in the cell. So I'm going to draw my cell here. And so I'm going to draw. So what we're going to do is we're going to build up a bunch of sodium out of the cell, because the pump that we have here is going to pump sodium out of the cell, and then it's going to pump potassium back into the cell. So we're going to build up sodium out of the cell, and then potassium inside the cell. Those are K's, if you can't tell. There's potassium inside the cell, and here's sodium outside the cell, okay? And so then this ionic gradient can then be used to kind of indirectly bring in other molecules. And so you'll see this. I'm going to show you, this is going to happen with a sodium glucose transporter. So again, we can use that ionic gradient established from active transport. So it's still considered a form of active transport, even though the ATP isn't used for this second step. It's used for this first step, right? But we needed that ionic gradient to be established. So then we could use that ion gradient to then derive the transport of another solute. So we'll talk about once we've established a bunch of sodium outside the cell, then you'll see we can use these sodium glucose symporters, and sodium just wants to move down its concentration gradient, and that symporter is going to bind to glucose with it and bring the glucose down with it, okay? I'm going to come back to the resting membrane potential. I'm not going to show you this video right now, but we will actually talk about resting membrane potential in just a little bit here. So sorry, I have this kind of out of order. So how we initially establish, I'm going to talk, since I'm talking about primary active transport, let's talk about how the sodium potassium pump works. Now, you don't have to understand all the details of this yet, but this is going to be very, very valuable to you at the end of the semester, so that you can understand how nervous conduction works. So, and it will also tell us how we can do, again, some of these secondary transports in just a minute by understanding this. So what ends up happening is this sodium potassium pump is really a pretty basic pump. Well, I shouldn't say that. It's actually very complicated, but we can kind of simplify it and make it a little bit easier for us to understand. And so if you can picture this sodium potassium pump just like it is here. Oops, go back. Oh, I'm not sure how that happened. There you go. So you can look here, and initially we can see that the sodium potassium pump has 3 binding sites here and 2 binding sites here. Okay. Now, on this side, when the sodium potassium pump is open to the inside of the cell here, okay? It's only going to bind sodium, okay, at this point. And so it needs to bind 3 sodium molecules, okay? When it binds those 3 sodium molecules, okay, then what can happen is an ATP molecule can then undergo hydrolysis and add a phosphate to it, okay? And the energy that's released during that reaction is what's really going to run the pump. So as it transfers the phosphate to this protein pump, the protein pump, it's going to cause this protein pump to change shape, undergo this conformational change. And when it does, it's going to pop open to the outside of the membrane now, okay? Releasing the sodium here to the outside of the membrane, okay? Now it's going to be open to the outside of the membrane, and now that's going to allow these potassium sites to kind of open up. And now the potassium sites are open. Potassium can then bind to the pump. And when 2 potassium bind to the pump, this is going to trigger the release of that phosphate. Boom, pop off the phosphate. And when it does that, it's going to undergo another conformational change and pop, release 2 potassium to the inside of the cell, okay? So what I want you to think of is, again, this sodium potassium pump is going to be open to the inside of the cell, okay? You're going to see here, you can picture this little triangle open to the inside of the cell. Boom, it's going to bind 3 sodium, then an ATP is going to come phosphorylate it, and then pop, it's going to open up, undergo this conformational change, and pop, open up to the outside of the cell, okay? Releases the sodium. When it does that, then it can bind to potassium. As soon as it binds that second potassium, boom, it's going to trigger the release of that phosphate, undergoing a conformational change, pop, 2 of those now sodium inside the cell, okay? So again, we're going to be binding 3 sodium, okay? And then with the help of ATP, pop, releasing the 3 sodium out of the cell, then binding the 2 potassium, then pop, undergoing that conformational change, okay, when it releases the phosphate, and bringing those potassium inside, okay? Now, this is going to help us establish, I'm just going to kind of mention this right now, even though I'm not going to get into this, but it will actually help us establish a little bit of a concentration gradient, or an ion gradient. It's going to help us establish a lot of a concentration gradient, because now we're going to end up with a cell. Let me draw this here. Sorry again, I feel sorry for you anytime that I'm drawing here, but I'm going to do my best. So what's going to happen, right, is we are going to be putting a bunch of sodium. I'm going to draw sodium big, okay, on the outside of the cell, because we're pumping 3 sodiums out, okay? And then I'll draw sodium as little on the inside of the cell, okay? So now we've established this concentration gradient where sodium wants to diffuse, right? It can't, it has to have a carrier protein to go through because it's charged, but it wants to diffuse down this concentration gradient. And then potassium, again, we pump 2 potassium in the cell. So I'm going to draw potassium big in the cell, okay? For every 3 sodium out. So we pumped 2 potassium inside the cell. So then I'm going to have a little, oh gosh, I can't get my pen to work here, a little tiny potassium out here, okay? And now potassium also wants to move down its concentration gradient, okay? It has to move through a channel protein because it can't just move through there, but it wants to move down that concentration gradient, okay? Now, the other thing that's interesting is that you're establishing a little, or I should say, we're gonna end up using this to maintain an ionic gradient across the cell here. And the reason is that you're pumping out 3 positive charges and only 2 positive charges back in. So that's going to help us establish what we call as our resting membrane potential. I should say maintain our resting membrane potential, where we end up with a situation where we're a little bit more negative on the inside of the cell and a little bit more positive on the outside of the cell, okay? And we'll talk about more reasons on why that is, but that gives you at least a little kind of hint as to what we're going to talk about kind of coming up here. Okay, so now let's see. I have this nice, oh no, it's not going to play my media. Well, I'll try to post this for you. I actually have the video that I have. There's a video I have. It's an awesome video on sodium potassium pumps towards the end of this lecture, and I'll show it to you. You can click on the link and watch it, and it's amazing. And it's a great little animation. So now let's get into secondary active transport, okay? So remember now that we've established this concentration gradient, lots of sodium outside the cell, more potassium inside the cell, right? So remember that sodium now wants to move down its concentration gradients. Again, I'm going to draw this where we have, because we've pumped 3 sodium out every time we use this sodium potassium pump, we have a lot of sodium on the outside of the cell, okay? And not a lot of sodium inside the cell because we've been pumping it out, right? So I'm going to draw a little sodium here in terms of its concentration, okay? And so what happens is then if you have these transport proteins, okay, that can, remember we talked about carrier-mediated diffusion. This is a case of that carrier-mediated diffusion. And this carrier is actually what we call a co-transporter, meaning it can transport 2 molecules. So what happens is it's going to bind to sodium, okay? And sodium, again, is just diffusing down its concentration gradient, but it can also bind a glucose molecule as well. And so what's going to happen then is as sodium is diffusing across the membrane through this transporter, it's going to bring a glucose with it. Okay, and that's what we call secondary active transport. Again, it doesn't directly need ATP for this second part, but it did need the ATP to establish that sodium concentration gradient that it's using. Okay, so that's called secondary active transport. All right, so let me exit out of my drawing there and move on to vesicular transport. And then we'll come back and talk a little bit more about using the sodium potassium pump in just a few minutes on establishing what's called the resting membrane potential. And again, hopefully I am hoping that some of this is a review, but if you are confused on this and you're just like, whoa, you lost me, please come see me. I say come see me. I know you can't see me in my office because I'm, this class is online, but I will set up some time virtually with you to go through this because I want to make sure that you understand this. It will really help you, again, not only just here in 201, but also carry through to 202. So this is an important lecture. Okay, so let's go ahead and talk about vesicular transport. And this is just the involving the transport of large particles and usually lots of them, okay? So it's either a really large particle or you need to transport, a lot of a certain particle, okay? And this is a way that we can transport things through these membranous sacs into and then also out of a cell, okay? Through vesicles. So this is kind of showing you this cell is transporting a microbe, which is a large particle, right? If you're talking about the cellular level here. And so a lot of times you can, again, transport that into the cell by trapping it in a vesicle, okay? And so this usually requires some ATP for this process, okay? So vesicular transport processes are going to include endocytosis, which is transporting things into the cell, and exocytosis, which is transporting things out of the cell. So you can think of the easy way to remember this is endo in, transport into the cell, and then exo, exiting the cell. You're transporting things out of or exiting the cell, okay? So think of the endo in and exo, exit, transport out of the cell, if that helps you out at all. There are 3 different types of endocytosis that we're going to talk about in just a minute. Phagocytosis, which is kind of what we consider cell eating. Penocytosis, which is cell drinking, but really it's kind of bringing in small particles. And then receptor-mediated endocytosis. And we'll talk about that's where you're binding very specific molecules, multiples of those molecules, and transporting them in a vesicle. And then exocytosis, again, is just going to be transporting things out of the cell. Okay. Transcytosis is just transporting things into, across, and then out of the cell. So when we talk, we won't really use this word all that often. I'll mention them here, but honestly, I'm not going to test you in transcytosis and vesicular trafficking. But just because it gets into a little more cytology than you guys probably need. But oftentimes we're transporting something into the cell, across, and then out of the cell, just like you saw here. So this would be kind of transcytosis. We're transporting something into the cell, across, and then actually digesting it, and then, transporting out of the cell via exocytosis. So we call that transcytosis. And then vesicular trafficking is just transporting from one area or organelle of the cell to another. So we'll talk more about vesicular trafficking and how we transport things from one area to another when we talk about transporting proteins from the endoplasmic reticulum to the Golgi apparatus and things like that. Okay, so again, endocytosis. This is probably a little more than this slide is a little more involved than, again, I think that we probably really need to get. But endocytosis is oftentimes you're going to bring things into the cell like pathogens. And we don't want those pathogens to kind of be running freely in the cells, so you can trap them in a vesicle. And again, once you trap those pathogens inside a vesicle, then you can fuse it with a lysosome, and that way then the lysosome, we'll talk about lysosomes a little later on here in the lecture, where they can actually lyse whatever that pathogen is that might damage the cell. But it's also, again, a way for us to just bring things into the cell that we need lots of, okay? So this is just giving you an example, and we'll talk about sometimes then we can actually do what's called receptor-mediated endocytosis in just a minute, where it's very specific things that we want to bring in the cell. Okay, so let's talk really quick about penocytosis. Penocytosis is really what we consider to be cell drinking, and it's basically bringing in fluid, but with small particles, okay? So small molecules mixed in it. So we call it cell drinking, but it's really eating small particles, okay? Where phagocytosis is what we call cell eating, and it's where we're engulfing large particles. So again, somebody early on thought they should call penocytosis because they thought, it's mostly like liquid because they couldn't see all the small particles. But it's actually bringing in small particles inside a vesicle, where phagocytosis is big stuff, you can think. That's bringing in large molecules, okay? And when you bring those large molecules in, again, you're going to trap it in a little bit of membrane here. So the membrane's going to kind of invaginate and pinch in, and then you're going to trap whatever that molecule, that large particle is, inside the membrane-bound vesicle now. And we call that little membrane sac a phagosome, okay? In this case, for phagocytosis, we call it a phagosome. And for penocytosis, not really important, they call it a penocytotic vesicle, okay? Again, just more, we have to give all these fancy names to these terms. But I don't really care that you know the name of the pinocytosis one, the pinocytotic vesicle. That's not as important as the phagosome, because you will hear us use the term phagosome later on in the semester. So phagosome is just, especially when you're talking about the immune system, when you get to the immune system and things like that. So phagosome is just when, again, a cell engulfs a large particle, or it can be a pathogen or a microbe, and you trap it and you pinch off a bit of the membrane, trap it in that membrane. Now it's in a membrane-bound sac that we call a phagosome, okay? So, and that phagosome, if it has, you know, some type of pathogen, can then, you know, possibly be digested when it fuses with a lysosome, okay, and ejected out of the cell, or, you know, if it's bringing it in, and can, you know, if it's you know, a molecule that the cell needs, it can then break down that molecule when it fuses with the lysosome and then release it inside the cell. So again, just a couple of things. Again, pinocytosis, cell drinking, is really more cell drinking where it brings in extracellular fluid and small solutes, okay? So keep that in mind. And then receptor-mediated endocytosis. I know, it's a mouthful, right? Receptor-mediated endocytosis. sounds more complicated than it is. All it is it's endocytosis that's mediated by binding a very specific solute to a specific receptor inside the cell, okay? So this enables the cell to ingest large amounts of a very specific substance, okay? And and again, it will bring that in and pinch it off into a vesicle. Okay. Exocytosis, again, is just where you're ejecting materials out of the cell. And this, again, is oftentimes activated by cell surface signals. So we'll talk about this in the nervous system when you have the axon terminal, and it's you know, from a neuron, it releases acetylcholine at its junction. It's going to get a signal to release that substance, again, out of the cell via exocytosis. So again, this typically happens when either the cell needs to get rid of a substance, like it broke down a pathogen, or again, it needs to release a substance or secrete a substance outside the cell, okay? So if it needs to secrete, let's say, you know, hormones outside the cell, then it will do this with a secretory vesicle that will eventually, again, fuse with the cell membrane and release that substance out, okay? So some substances are exocytosed, like hormones, neurotransmitters. Also mucus can be released this way, sometimes cellular waste. Those are all things that can be released by exocytosis. So we'll talk a lot about exocytosis when we, especially, you know, things like the endocrine system, where we're releasing hormones, you know, from different organs. So again, or even in the digestive system, we'll talk about, you know, how cells can release digestive enzymes via exocytosis, things like that. Okay, so, and this is, again, a little more than I think you probably need to know, but there are, just in case you hear of them in the future, I'm not going to test you specifically on T snare and V snare, but there are these molecules that are bound. There's the T snare molecule that's bound in the plasma membrane, and the V snare molecule. This is a protein at the surface of that vesicle that's going to secrete a substance, okay, via exocytosis. And so that what ends up happening is that V snare on that V snare protein on the secretory vesicle will bind to the T snare protein on the plasma membrane. And once, again, those proteins fuse, then that will literally allow this portion of the plasma membrane, okay, to, oops, let me go back here, then now fuse with the outer membrane of the cell there, and then release that substance. And then this membrane will then just become part of the exterior of the cell. So as things are transported, okay, throughout the cell and vesicles, the membrane that's surrounding those vesicles is made up of the same stuff that your plasma membrane is. So that way then again, as things are brought in and out of the cell, we're just kind of like basically recycling the membrane parts as we transport them around throughout the cell. So again, that's exocytosis. Now, the last thing that I want to talk about here is membrane potential for today. And again, that's because we already kind of talked a little bit about this. Again, I'm not going to get too much into this because we are going to talk about this when we get to the nervous system, but I like to mention it now just because I figure the more I can get these concepts into your head, the more likely they are to stick. So when we talk about resting membrane potential is this idea that there's a separation of charges right across the membrane, and they're opposite charges. And so in the case of the resting membrane potential for cells, we're going to end up with a negative charge on the inside of the cell and a positive charge directly outside on the other side of the cell membrane. And there's this difference in that electrical charge, okay, across the membrane that we're going to call voltage, right? The difference in any electrical charge between 2 points is called voltage. And so this voltage difference across the membrane can actually be measured, okay? So cells, again, are charged, but really primarily they're charged just at the membrane, okay? So if you were to mix, if you looked in the inside of the cell here, on the inside of the cell here, the positive and negative charges are actually balancing each other out usually. So it's more neutral, except for right along the membrane. It's right along the membrane that you're going to get this buildup of negative charges, and right along outside the membrane that you're going to get this buildup of positive charges, because those positive charges are going to be attracted to the negative charges on the outside of the membrane. Okay? And this makes our cells what we call polarized. Okay? So they have this polarized potential. And so when we talk about this, if we were to measure the voltage distance, or the voltage, across the cell membrane, which we can do, typically the membrane voltages range anywhere from -50 to -100 millivolts. And what that means is that negative sign tells you, it indicates that the inside of the cell is more negative, okay? So it's just measuring that difference in electrical charge across the cell, and the negative indicates that it's more negative on the inside, okay? So let's talk a little bit really briefly. Again, this is a little more than I want to get into. This video is a really awesome video that you can watch, and it is at the level. So go to my PowerPoint, please watch this video. I don't have time to show it to you, but it is a great video, and it shows you at the level that I am expecting for this lecture. So again, your book gets into a little bit more in terms of setting up this electrochemical gradient, but active transport is going to help maintain our electrochemical gradients across the cell. And it's going to do it with, you know it, the sodium-potassium pump, right? That's why we talked about it, and that's why I used it as an example for active transport. So when we talk about, we have established kind of this gradient with that sodium-potassium pump, right? Where we have lots of sodium on the outside, lots of potassium inside the cell. What's going to happen is we do have these potassium leakage channels, and some of the potassium is going to diffuse back out of the cell through these potassium leakage channels down its concentration gradient. Now, so that's a positive charge that can leave the cell, right? So let me draw this here. So here we have the cell, and we do have some of these potassium, not all, we're still going to have our concentration gradient established, but some of these potassium are going to leak back down their concentration gradient out through these leakage channels, okay? So remember, these are going to be channels that are not gated. They're just leakage channels. There are gated potassium channels as well. We're not going to talk about those in this lecture, don't worry. But there are also leakage channels. And so some of that potassium will leak down. So you have a positive charge leaving the cell. But here, now the challenge is that negatively charged proteins can't leave. So they stay in the cell. And so you build up around the membrane these negative charges, okay? And those are really your anions and especially your proteins, your negatively charged proteins, okay? And so as a result, the cytoplasmic side of the membrane, as some of the potassium leaks down and then we're left with these negatively charged proteins that can't leave, okay, we end up with this negative charge on the inside of the membrane. Now, in many cells, sodium can also leak down its concentration gradient. Because remember, we have lots of sodium from those sodium-potassium pumps, right? We have lots of sodium on the outside of the cell, and it can leak down its gradient. But the challenge here is that the membrane has far fewer sodium leakage channels. So what happens is the membrane is much more permeable to potassium than it is to sodium. So really, there's not a lot of sodium leaking back in. There's potassium leaking out. So we got positive charge going out, and we're left with these negative charges on the inside that can't leak out of the membrane. And so this is a very complicated way of stating that this is how we get the electrochemical gradient, okay? And so what I really want you to know is that really potassium is actually what establishes this electrochemical gradient of it being more negative on the inside of the cell, because that potassium can leak out and the negative ions cannot. The negatively charged proteins, negatively charged proteins and ions cannot. So what that means is we have positive charge leaving, negative charge stuck on the inside of the cell, and not much positive charge coming back in because the membrane's more permeable to potassium. Okay, so this leaves us with this negative charge on the inside. Again, most cells have a resting membrane potential of around negative 90 millivolts. So again, this is just a very great video. If you can click on this video here, it is an awesome video on the sodium potassium pump and the resting membrane potential. And it's by the Amoeba sisters, and it is great. If I had time to show you the video, it basically summarizes everything that I've just talked about in terms of resting membrane potential, but it does it with these great animations in a way that just is awesome. So please take the time to watch this video, especially if you're struggling with this idea of establishing the resting membrane potential. So again, this is just showing you what happens with potassium, helping to establish that membrane potential by leaking out, right? And then some potassium, again, is also going to be leaking back in and attracted to those negative charged ions here. But again, we have more potassium leaking out than we have sodium leaking back in. So you can see, again, it's really going to help establish this negatively charged interior of the cell, okay? And again, what helps to maintain that electrochemical gradient, right, is we have to maintain that sodium-potassium gradient across the cell. And we do that with that lovely sodium-potassium pump. So remember, that sodium-potassium pump is going to pump 3 sodium out, 2 potassium back in. So again, that sodium-potassium pump is just, it's so important to understand because it will really help you out kind of throughout the semester. But remember, that sodium-potassium pump is going to be open to the outside here, or open to the inside of the cell here. It's going to bind 3 sodium. ATP is going to phosphorylate it. It's going to pop, okay, and pop open to the outside of the cell, releasing that sodium to the outside of the cell, right? And then it can bind 2 potassium, right? And when it binds those 2 potassium, it's going to pop back open to the inside of the cell when it releases that phosphate group, and boom, potassium can then come into the cell. So it's going to, remember that this sodium potassium gradient here, where sodium builds up on the outside and potassium on the inside, is really established by this sodium potassium bump that brings 3 sodium out of the cell and 2 potassium back in the cell, okay? And that's what helps to maintain this steady state of this resting membrane potential of the cell. So we'll talk about when we get, again, to the nervous system and the muscular system, how neurons and muscle cells actually upset this steady state of resting membrane potential with gated, excuse me, something caught my throat there, with these, excuse me, 1 second here. Just like a real lecture with me, right? So what happens is in neuron and muscle cells, for a neuron to fire and muscle cells to contract, what happens is to start that process, they have these gated sodium and potassium channels that will, again, be triggered to open and upset that resting membrane potential, okay? So that's why it's so important to understand resting membrane potential. So here's those lovely, I have these awesome memes on resting membrane potential, to 70 millivolts and beyond for resting membrane potential. And your neurons must be firing. I sent action potential. So that's, again, what we're going to talk about when we upset resting membrane potential in neurons and what's called an action potential. And again, this is just that example of secondary transport using those symporters. Okay, so again, if you have any questions on this, then please ask me, and I would love to explain this. I know that this is very involved, and so and I feel like I'm still doing this whirlwind of just giving you this little bit. There's so much more, believe it or not, that we're leaving out, and that's what makes this so challenging, is that I'm kind of having to take this like trip around the world, right? And for the United States, I might show you, you know, Phoenix, and I might show you New York, and everything else in between I have to leave out. So if you, again, are struggling with this, please come see me before it becomes a problem. Okay, the next lecture, sorry, this one went a little long. The next lecture, I'll try to keep it a little shorter, and we'll start to actually cover stuff inside the cell. So this is just a quick thing on membrane receptors, but then we're going to get into the nitty-gritties inside the cell. Can you believe that we've only been talking about really what's happening at the plasma membrane? That's how important it is. So again, if you need any help, just contact me via email. Through Canvas is the best way to email me.